Hello students, today we are going to study the mechanism of action of enzymes. The Michaelis-Menten theory is the basis of enzymes mechanism of action. That is, enzyme plus substrate, it gives the enzyme substrate complex, and this enzyme substrate complex then afterwards complete the reaction and produces enzyme and products of the enzyme action. During the formation of ES complex, the substrate molecules attach at specific point on the enzyme molecules and are known as active sites. This is given by two theories of mechanism of action of enzyme. First one is lock and key theory and second one is induced fit theory. In this lock and key hypothesis, the substances on which the enzymes acts are called as substrate. Enzyme bind to the substrate by the lock and key hypothesis. This theory was proposed by Fischer in 1894. It is like a lock and key. In this case, the shape of the active site of enzyme and that of the substrate is complementary to each other. The substrate molecule fits into the active site of the enzyme just as the key fits into lock, hence called the lock and key model. The shape of the active site is rigid and the complementary to the shape of the substrate complex. It is shown in this figure. This enzyme reaction depends on the presence of active sites. Active sites have a specific 3D shape. This specific substrate molecule fit into the active site like a lock and key. The substrate bind to the enzyme forming the enzyme substrate complex, and it is convert the substrate molecule to the product molecule, and these product molecules separate, leaving the enzyme molecule unchanged and free to combine again with the more substrate molecules. Next is the induced fit theory. Current hypothesis for the enzyme action: when the substrate molecule fits into an enzyme molecule, the enzyme molecule alters its shape slightly so that it fits more tightly around the substrate molecule. This makes a chemical reaction easier. This theory was proposed by Koshland in 1958. In this case, the shape of active site of an enzyme is flexible so as to accommodate a wide variety of substrate molecule. Substrate induces conformational changes in the enzyme. The shape of the active site of enzyme is made complementary to the substrate molecule. It is shown in this figure. Both hypotheses, lock and key hypothesis and induced fit theory, it is shown here. See what is the difference between this lock and key mechanism and induce the fit theory. Lock and key mechanism, this theory is proposed by Fischer in 1894 and induced fit theory is proposed by Koshland in 1958. Second difference is active site of the enzyme in lock and key mechanism is rigid and active site is flexible in induced fit theory. In lock and key mechanism, only one kind of substrate can fit. In lock and key mechanism, only one kind of substrate can fit. And in induced fit theory, variety of substrate can fit. And in lock and key mechanism, substrate cannot change the enzyme conformation. But in induced fit theory, substrate induces conformational changes in the enzyme. This is all about the enzyme mechanism by these two theory. Next lecture we will see enzyme inhibition. Thank you.